Thanks everyone for checking in today, and we're going to take about, I would estimate, probably 30 minutes to 40 minutes just to give what I would estimate to be a very high-level introduction to bioinformatics. So there's very few assumptions made about people's background knowledge. So this is meant to be educational and just people interested, like what is this all about? This should give you a bit of an introduction and a little bit of background just to get you started. I'm going to move on here. Okay, so before we get too deep into what bioinformatics is, I just wanted to do a little bit of an introduction of what we do here at Canada's Michael Smith Genome Sciences Centre in beautiful Vancouver, British Columbia. So we are part of the BC Cancer Agency, and as you would expect, being part of the Cancer Agency, we are very involved in the research and trying to help ways to improve the lives and outcomes and treatments for not only cancer patients, but patients with other diseases. So my job, actually, I just looked it up, it was almost exactly 12 years ago to the day I started working here at the Genome Sciences Centre. I started here some with a background in engineering and computer science and not necessarily specifically in bioinformatics. So that's one of the great things about this field is people with different specific areas of knowledge can contribute, and as you're working in this field longer and longer, you just start to learn all the other bits and pieces that go along with it. Okay, so I'll move on to the next slide. This is where I'm going to actually start to talk about what, in fact, bioinformatics is. So here at the Genome Sciences Centre, there's about 300 employees working on the research projects that we have. I sent out a survey to all the people who are, in fact, professional people doing bioinformatics to ask what words come to mind if you had to describe bioinformatics. Here's a word cloud that I put together just with all the responses that came in. I thought it was really interesting, actually. It worked out almost perfectly because just the order things showed up here on the slide, you can almost read off computer biology research. And that, there's all these other words, statistics, engineering databases, genes, genetics. They're all related, but I think if you had to distill it down just to a few topics to describe bioinformatics, you're talking about using computers. Those are the informatics part of bioinformatics, and then of course you have biology. So with my engineering background, I try and describe things in terms of inputs and outputs. So if I don't know how to describe the thing I'm actually talking about, I attempt to do it just by talking about what are the things going into it and the things coming out the other side. So bioinformatics, as most of you likely already know, is a very broad field. And some of the things that people doing bioinformatics would study are sequence information, sequence data, and we will spend quite a bit of time talking about sequence data. There's other experiments like array experiments. Something that we used to do a lot here is run lots and lots of electrophoresis gels. And another part of bioinformatics can just work on computational models without actually working on data as much as the theory behind the algorithms. And if bioinformatics is successful and things are working the way we want them, we will get scientific publications. We will help people that need help in terms of being sick and need to find cures. And we'd also make environmental discoveries by applying the algorithms and resources that we have. Because bioinformatics is so broad, I won't be able to talk about the whole field of bioinformatics in such a small period of time, and I wouldn't consider myself qualified to talk about the whole swath of everything that would qualify as bioinformatics. But the next 25 minutes or so is going to be focused on talking about how we use sequence data in bioinformatics to generate things like publication and to improve patient care. Like I said in the introduction, we're not making much assumptions or many assumptions about what people understand. So I just wanted to, for anyone who doesn't know what this is all about, just a very easy background. So when we talk about sequencing, genome sequencing in particular, so we don't have to, but we often start with cells. The orange things in the cells could be chromosomes or genomic DNA. We're going to extract that DNA from the cells, and then we're going to chop up the DNA. And In the ideal world, we wouldn't have to chop up the DNA. We could just read the sequence right off the chromosome. And there are newer technologies that do this sort of thing. But the most efficient and the highest quality data that we can find right now, we have to apply, we extract the DNA, then we shear it into smaller pieces. We either sonicate it or use chemistry to do that. Then we take these smaller fragments of DNA and then we attach adapters onto the ends so that we can do the sequencing experiment. So we take this almost test tube full of these smaller fragments of DNA. They're usually on the order of 100 to 500 base pairs in length, load it on the sequencer, and soon after that is when actual bioinformatics takes over. So the sequencing that happens on the sequencer is like a digitization process. So it's taking stuff that is actually happening in the real world and then reading out data to us where we start to see sequence reads. So off of this sequencer, we see reads usually on the length of 100 to 200 bases. I've showed some very example sequence down here on the bottom right-hand corner. These are cartoon sequences. Usually they're much longer than this and more interesting, but this is really what we're focusing on when we're talking about doing bioinformatics at the Genome Sciences Center. So here at, I'm going to say GSC, which is the acronym we use for Genome Sciences Center, we have a number of sequencers. So we have 5 HiSeq X sequencers. These are some of the newest technology. We have HiSeq 2500 sequencers, NextSeq, and MySeq. So all of these are made by Illumina. You could read about Illumina sequencing if you're interested. But what I want to point out here is that when all of these things are running, and it is possible to run all these in parallel anytime if we want to load them all one day, they'll all be running at once, we can generate 55 million bases of sequence every second. So that very long sentence I just spoke, you could get hundreds of millions of bases sequenced in that amount of time. To put that in perspective, the human genome is about 3 billion bases, give or take, depending on your perspective. At the Genome Sciences Center, we can sequence the amount of sequence that's in the reference genome in about a minute. And just for comparison, we can talk about the reference genome that it took roughly 10 years to put together and many, many people, many, many millions of dollars put together to do that project. So this is the crux of the bioinformatics problem that we're working on here. So we are, if we're sequencing say a genome for a cancer patient, we are going to sequence about 2 billion reads of sequence. Each of those might be 150 bases long. And we want to interpret that information in a way that we can find some use in it in this huge ocean of data. And one way we do that is by comparing it to a 3 billion base reference genome. So we're looking at lots of zeros here, even if you're not into computational analysis. If we're taking 2 billion things and each of them is 150 little measurements, and we want to somehow interpret that with respect to 3 billion other observations, this is a very computationally complex situation. Okay, so this is, I think, if you haven't worked in bioinformatics, you may find this very interesting. So when we do all this sequence, you could think about publishing an encyclopedia for everything that we sequence, where we just write every base on every page that we observed, when in fact it's way more efficient and way more interpretable for us to compare the things that we sequence to a reference genome and then describe our sample just in terms of how it differs from the reference genome. So when we do a whole lot of sequencing, we look at all this data, we usually end up with a few relatively small files at the end that just say, how is the sample that we are sequencing different from some other samples, or different from some other reference genomes? When we do this, of course, if we're publishing things or sharing information with people, we have to both share the differences that we observe and also the information for how to acquire the reference genome that we're comparing against. So this is sequence read alignment. Just in very basic terms, so we're sequencing these 2 billion reads, And we want to know from which part of the genome each of these reads originated. And this is an easy to understand computational problem in terms of we have a bunch of strings with A, C's, T's, and G's in them, and we want to find where each of those strings will match in the reference genome. When we do this operation, it's called alignment. There's lots of tools that are available online if you had your own data that you wanted to align to a reference genome. The thing is that this, although is a simple thing to understand, when you're looking at billions of reads against a multi-billion base reference genome, this is computationally complex and takes a whole lot of computing horsepower. So what does this look like in real life? So back on this slide, this is of course all cartoons, and the sequence reads are much longer than this in real life. So this is a screenshot from a tool called IGV. IGV is really commonly used for people when they're working in this field because it allows you to look at all of these billions of reads. You can zoom into a particular region of your reference genome and observe if you're seeing what was in the reference genome or if your sample differs in some way at that location. So a couple of things I want to highlight in this IGV screenshot is on the bottom here, going across, you can read T, 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 A, 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 A. This is the reference genome. So we've at this point zoomed in on, it's written up here, chromosome 15. It's pretty small on my screen. So I think it says chromosome 15. And this is a cartoon, if you will, of chromosome 15 with the centromere. And we're looking at this very, very tiny spot on chromosome 15 where this red mark is. And at that location, we see a window of about 50 bases. Each of these horizontal gray rectangles with arrows on the end, these are reads that have been aligned to the reference genome. When a base is in one of those reads that doesn't match the reference genome, it'll be shown on this sort of plot. So we can see over here there's a C highlighted, and that C I think is lining up where there was an A in the reference genome. When we see a single C like that, it's often something that might be an error that happened during library construction or on the sequencing instrument. However, we do deep sequencing so that we can determine which of these mismatches stack up and statistically give us some confidence that our sample actually differs from the reference genome. And what we're showing here right in the middle is this one position with this big stack of C's where the sample that's been sequenced here differs from the reference genome. Interestingly, this variant and a couple other variants in combination would determine that this particular individual has been sequenced has a 90-ish% chance of having blue or green eyes. So read alignment is fantastically useful. We can interpret and find wonderful results just by aligning the reads and looking at the differences to the reference genome. However, there are lots of situations where 150 base read maybe doesn't give you enough sensitivity. You can imagine in a disease like cancer where there's many bits and pieces of chromosomes that might join together. So you have disparate regions of the genome joined into one continuous piece. You need a lot of sensitivity so that you can anchor different parts of your sequence. So what we do in this case is what's referred to as de novo assembly, where we take, rather than taking each of the reads and aligning them to a reference genome, we take each of the reads and compare them against each of the other reads that have been sequenced. This is obviously more computationally complex than the alignment problem because here we're taking every read and comparing it to every other read. But what this gives us, we construct these things that we refer to as contigs, which I've highlighted here in green, where we are able to take each 150 base read, stack them together to build a continuous sequence that's longer and perhaps more confident than each of the individual reads that make it up. Then we can take those contigs and align those to the reference genome, and those will allow us to detect what we refer to as structural variants, and they're often more complex and harder to detect things in terms of what we see in our sample that's different from the reference genome. This is computationally complex, so you can, depending on your application, you may decide to do this or you may decide not to, depending on what resources you have available and what sort of things you're looking for. Okay, So far, we've just introduced some of the very high level, not really how anything works, but just the sort of things that we do in bioinformatics here at the Genome Sciences Center. And just a couple of points I wanted just to go over again to make sure we're all on the same page. So, of course, as I mentioned, we sequence lots and lots and lots of sequence here. And of course, we need lots of resources to be able to deal with such a large amount of data. We do alignments and assembly of the reads. We do different approaches depending on the specifics of what we're trying to find. And importantly, when we report, we don't report every base that we sequence, although there are cases where we would, for research purposes, share every base that we sequence. But if we're describing a sample in terms of results, we only report the results and how our sample differs from other samples. That, in effect, is like a compression algorithm where we only have to use certain small amounts of data to describe the entire sample that we're looking at. So now we're going to change. What are we going to change? Well, we're going to refocus, I guess, in a parallel theme. So everything I've talked about so far, we're more or less talking about DNA sequencing or genome sequencing. Here at the GSC, of course, we can produce a number of different types of data. I've listed a few here. This is by no means exhaustive. We do a large number of other types of experiments, but here I just listed epigenomics. So here we're talking about sequencing, not the DNA, but the things around the DNA that may control important function in the cell. Proteomics, of course, we can identify and do some analysis of what proteins we observe in cells. Exome and capture type experiments. So rather than sequencing an entire genome, these types of experiments, you can just target certain parts of the genome. Of course, if you're only interested in one or two genes, then maybe you don't want to pay to sequence the whole genome. And then RNA sequencing, which I'll talk a little bit more over the next couple of slides, allows us to query and find results in terms of RNA expression in the cells. So this is back to high school biology, and I like to go back and review some of this stuff myself from time to time. So we talk about the reference genome, and I've drawn the genome in an orange squiggly line because it's super long and it goes on and for very far distances in each direction. Encoded on the reference genome, we have different very important regions that are often the focal point of research that we call genes. So I have 3 genes drawn here. They're in different places along the genome. And if we try to compare what we're getting from the genome and the transcriptome, it's important to consider that in the genome, we're measuring whether or not something is present. So we're going to measure if there's a SNP, single nucleotide polymorphism, just like that C we saw when we were looking in the IGV screenshot earlier. We might find copy number changes. But we're more or less describing the genome in terms of what is there or not there. So a binary observation. When we're doing transcriptome sequencing, which is a big word to describe RNA sequencing, we're doing it for the purposes of quantifying how much of each gene is present in our sample. So here on the slide, I've shown 3 genes again, but I've shown that each of the genes can be present at different amounts within a sample. And this is well, there can be philosophical discussions about what's more important. Is it sequencing RNA or DNA or maybe something else entirely? But we do show, and I will show coming up in a couple of minutes, how we combine RNA and DNA to help us get more information and hopefully more accuracy in the results that we're providing. Here I just want to summarize this. So if you're new to bioinformatics, you may or may not have heard of some of these things. So we sequence the DNA, we can get more SNVs, we can get copy number changes, we can get structural variants. There's other things that we can find. But the important thing to note here is we're talking about things that are either present or not present. We are not talking about them in a quantifying sort of way. On the other hand, we can sequence the transcriptome by accessing the RNA in the cells. And here we're just looking at how much of each gene is present. We can also measure or observe gene fusions. And we can, although it's not necessarily recommended just to sequence RNA to find SNVs, but we can also, because we are taking RNA reads and aligning them to the genome, we can find small SNVs in RNA libraries as well. Okay, so now I'm going to walk you through, in the next couple slides, just a very high level of how bioinformatics is applied to personalized medicine here at the Genome Sciences Center. So if an individual is unfortunate enough to have, say, melanoma, in this case, in my example, we can, in some cases, access the samples from the patient. So we take both a tumor biopsy, and that goes off to the right in my figure, and we can get normal tissue as well, and that might be normal skin, or it might be from blood from the same individual. By doing it this way, we can compare those two samples and subtract what's in the normal from what we observe in the tumor to find the specific somatic variants that might be driving the cancer in this individual. When we go through this experiment, we sequence, what I have on the left is 40x genome coverage of the normal sample. So I haven't talked too much about this, but when I say 40x, we're talking about, on average, every position in the reference genome having 40 reads covering it. When we sequence the tumor, we tend to sequence it much deeper. So in the example here, I've said that we sequenced the tumor genome to 80x coverage. That allows us to identify smaller or things at lower prevalence in the tumor sample. We want to make sure we're not missing very important things. We also sequence the transcriptome library. So here we're going to identify SNVs and copy number and things from the genome, but we're also going to identify maybe oncogenes, or maybe we'll find a certain pathway in these cells is highly regulated, which would help us inform the clinic that maybe a certain drug would be appropriate for this patient. All of this is done by the processes that I've described already. So we talk about alignments and assembly. All that stuff is happening down here in bioinformatics. We take the assembly results, we take the alignment results, we compile everything together, and we get results that look like this. So on the top left, I'm showing an example of what we call a somatic SNV. So in this figure, there's two samples represented in IGV. On the bottom, we have what we call the normal for this sample. So the sample that goes to the left up here. And on the top, we have the tumor. Like we were talking about before, when we see colored letters, and you can't see the letters because this is too small, so they're just little red bars, but these red bars indicate that within the tumor at this location, we see a non-reference base that is not present in the normal. So we have a somatic variant in the BRAF gene. This is a known canonical cancer variant. It's BRAF V600E. In this patient, this would be very interesting, and we could determine and feed that back to the clinic that we see this very interesting observation that might help them decide a better way to treat the patient. On the right, I'm showing what we often do with our RNA sequencing to QC things before we do a lot of interpretation. So what we do is we have all this downloaded data and previously sequenced data of RNA transcriptomes. And what we can do is correlate the expression of all the genes in the samples that we sequence with the expression signatures of all the published different data types that are available online. This allows us to do a couple things. One, of course, is to confirm the diagnosis. So if we expect to see melanoma, or we think we're sequencing melanoma transcriptome, we can correlate the expression of the genes that we observe in our melanoma sample and see if it looks like previously published melanoma samples. Sometimes we think we're looking at melanoma, but we're actually looking at something else. And this sort of correlating the gene expression can help with that. It can also, by a correlation, help us determine subtypes. I guess a classic example is if we're looking at breast cancer, there's certain subtypes of breast cancer that are easily identifiable by looking at overexpression of just a few genes. On the bottom left, this is more information that we would pull out from the transcriptome. Here we're looking at gene fusion analysis. So we've done a de novo assembly using the RNA reads, and then we take the contigs generated from the assembly and align it to the reference genome. That can allow us to detect gene fusions, which are, of course, of high interest in cancer research. On the bottom right, I'm just showing an example figure of somatic number. Down here, the colors of the dots represent the interpretation of what's happening at that location. So where we see a blue bar on the left hand of this figure, that would indicate a copy number neutral part of this chromosome. As we get close to the centromere, where the sequence gap is here with no data, here we see a copy number loss, and then the Q arm is gained. So we can take all of this information and collect it together. So when we're doing personalized medicine or personalized oncogenomics that we do here, we do all the sequencing for patients that, of course, need our help. But when we take the genome and we take the RNA and combine all the analysis approaches that we have, we can take a bunch of SNV calls and outliers in terms of gene expression and copy number results and collect them together to provide a report which will list a number of if there are therapies that might be recommended based on the variants that we see. We have had a good, a satisfying level of success by feeding this information back to the clinic. And this is the sort of thing that we're aiming toward with the bioinformatics. So all those analytical approaches that I'm talking about, we can do all of that stuff, but it definitely means a lot more if we can get down to the end and we can start to see differences happening in the clinic. So we're getting down to the end of what I wanted to talk about today. In summary, I thought about this for quite a while to try and think of, like, as an introduction to bioinformatics, how would I describe, like, what is this bioinformatics? You know, how do you describe it in just a few words? So what I came up with is bioinformatics is, and then I put a little disclaimer in there because I'm sure different people have different ideas, the process through which the interpretation of billions of sequence observations yields a distilled list of actionable findings. So all the things I talked about, alignment and assembly, doing copy number analysis, fusion analysis, all of this stuff is basically taking oodles and oodles of data and finding the very specific events that we can report back that can be used. And then I just listed at the bottom, bioinformatics is definitely not something that can happen in isolation. It has to have lots of support from different parts. Here at the Genome Sciences Center, we're really lucky to have a very powerful computational infrastructure. The people at Westgrid who are supporting this webinar, of course, have their own infrastructure that they can share. There's a very rich field of algorithm development devoted to bioinformatics. And of course, to do all of this stuff, we need to combine a number of trained individuals with different backgrounds so we can come up with the best results. And I think that's the last thing that I had planned to talk about. So I think now, if anyone has any questions, I'd be happy to entertain them at this point.